So, how do we get to this stage of tiny pieces of animal tissues in these 2 ml Eppendorf tubes? Let us take a trip and see where this interesting journey begins. How we get to the stage of animal tissues being deposited into our biobank, the relevance of why we do this, and how it could benefit our wildlife populations and the environment around them. The journey begins in the field, beautiful and sometimes difficult to access locations, where researchers travel to collect specimens which meet their specific scientific research aims and objectives. When specimens are collected, a tissue sample is taken, generally a small section of muscle tissue or fin clip, and deposited into our biobank. So, what exactly is a biobank? A biobank is similar to a bank or library, but instead of collecting and storing money or books, we collect and store biological tissue samples of animals and their accompanying information in a systematic way for scientific research purposes. At the National Fish Collection Facility at the South African Institute for Aquatic Biodiversity, we specifically collect and store fish tissue samples in our biobank. By researching the sequences of the base pairs in the DNA of these samples, we can discover a great deal about an individual as well as their species. Storing of our samples at the biobank is essential so that scientists can generate DNA sequences that are accurate and reliable. In order to ensure this, samples must be stored at very low temperatures to prevent damage to the DNA. This can be as low as -80 degrees Celsius, which is the temperature we store our samples at. We have multiple -80 degree freezers which store a large number of samples. When storing large numbers of samples, it is important to keep them well organized so they can be located easily. To help us do this, each sample is stored in a uniquely labeled tube. Information related to the specimen the tissue was collected from is stored in a software called Specify. This allows researchers to search for and request tissues from specific species of fish they are working on. So, why do researchers need our biobank samples and how will they be used? Unfortunately, many species, both aquatic and terrestrial, are close to extinction. Threats and challenges faced by species populations include climate change, habitat loss, and poaching or overfishing. This makes it difficult for species to survive in the wild, and numbers are drastically decreasing. Researchers can use biobank samples that have been collected over the years and across the globe to understand a species' genetic diversity. High genetic diversity within a species population helps improve resilience to the threats faced by that population and helps them to evolve and adapt to changes, increasing their likelihood of survival into the future. Genetic profiling can also help us improve how we manage captive populations in zoos or aquariums to keep them healthy and thriving, as they may need to be reintroduced into the wild one day. So, from all this information, these tiny tubes stored in our biobank contain a lot of important samples and information and can help in conservation efforts to protect and sustain our wildlife for future generations.